Bismillahirrahmanirrahim. Assalamu alaikum and good day everyone. Thank you very much for joining us today. I am Hazima Mozam and I welcome you along with my colleagues Dr. Hena and Dr. Humaira from ICCDS in the 11th lecture in the series of Comstech Distinguished Scholar Program. And we are very delighted and honored to have Dr. Ilkay Orhan with us today. Thank you very much, Dr. Ilkay, for your time. And Dr. Ilkay Erdogan received her PhD in 2002 from Gazi University in Turkey. She was visiting scientist at University of Winnipeg, Canada. Currently, she is Dean of Faculty of Pharmacy at Gazi University in Turkey. She has authored and co-authored more than 247 scientific papers, 18 book chapters, and 3 books. And she has 4 patents to her credit. Dr. Orhan has received several scientific awards, including Comstech Award for Science and Technology in 2010, OWSB Award for Young Women Scientist, again in 2010. She is also a recipient of Young Female Scientist Award by L'Oreal and Turkish Academy of Sciences. Her research interest includes work on phytotherapy, enzyme inhibitors from natural sources, neurodegenerative diseases, and natural cosmetics. Today, the topic of our lecture is plants and their metabolites as leading molecules for pharmaceutical industry. Dr. Ilkay, once again, thank you very much for giving your time. I hope our listeners will enjoy your talk and benefit from it. Over to you, Dr. Ilkay.
Okay, thank you very much, Fazima, for the nice introduction. I hope everybody can hear me clearly. So shall I start sharing my screen? Is that fine? Okay. Just a second. So can you see it?
Yes, Dr. Ilkay, we can see your slides.
Okay, then I can start.
Yes.
So thanks again for the nice introduction, and I would like to say hello to everybody, all participants from Ankara. So today I will talk about plants and their metabolites as leading molecules for the pharmaceutical industry. So actually in the first part of my talk, I will just give examples about the molecules that are already in clinical use, which have been served as the drug leads or clinical available drugs. And in the second part of my talk, I will just go through my recent studies. So oops, it doesn't go. I don't know why. Sorry, I cannot just go forward. Okay, now it's fine. So as you remember from my CV, I'm from Pharmacognosy department actually, and I'm working on natural products. So pharmacognosy basically deals with the drugs or drug raw materials from natural or we can call biological sources. So that means we use any biological source as the research materials that of course consist of mostly plants, and then of course animal sources, marine organisms, and microorganisms as well. All these biological sources are within our scope. And of course we benefit biodiversity a lot. And as you can see here, the world has a big biodiversity, especially in some parts of the world. And looking for an effective, safe, and clinically available drug lists from natural sources or biodiversity, it is, kind of digging a haystack, looking for a needle. So this is a very tough work, as you can appreciate, and this means we need to work a lot to reach at an appreciable success. So this is a great work, and you really have to enjoy it. So the first drug that I would like to give an example is Epibatidine. So this was actually a natural product initially isolated from a frog species called Epibatopathes antoni. So as you can see, this is a very, very small frog species, almost equal to size of a coin. But this molecule was isolated in 1992 by John William Daly, who was an American scientist, through a research grant provided by NIH. And then they, discovered this drug, Epibatidine. And then this was a kind of model compound. Actually, it was developed by Abbott Drug Company, and then its several derivatives like terbinafine or abinafine, they were also synthesized through modeling this drug. So this has a capacity to bind to nicotinic acetylcholine receptors, and it has a great memory enhancing effect. Besides, it has great analgesic found that it has 200 times stronger activity actually
Well, I think
than that of morphine. So another example is Captopril. This drug is also very well known and especially used for hypertension patients. And it was actually modeled from a natural product, and it has a very interesting discovery story behind that, because the British pharmacologist John Wayne discovered this prodrug, maybe I could say, while he was working on drug candidates effective against hypertension. And in meantime, in his research group, actually, there was one guy joined from Brazil, and that was Sergio Ferreira, and he brought a venom extract with him to England, and then they found out that it has a great actually antihypertensive activity. So they discovered this BPF factor actually, and at that time actually John Wayne was consultant to Squibb Pharmaceutical company, and he mentioned them about this molecule having great ACE inhibitory activity. And then Squibb headquarters actually they supported R&D work of this molecule, and then it was developed into a clinical drug, and Squibb made a lot of profits, like billions of USD. And but before that, for the clinical trial stage, UK didn't let the researchers to carry out clinical trials within this country. Then they took the molecule to USA and they did the clinical trials, and then they got FDA approval in 1980s. And this work also brought a Nobel Prize to John Wayne in physiology and medicine. So a natural product also can give you definitely a Nobel Prize as well. So another drug developed from a natural product that I would like to mention is exenatide. This was also very, very big molecule, as you can see here, a peptide derivative molecule initially isolated from the venom of Gila monster, which is a Helioderma suspectum. So this creature's venom actually afforded this compound, and the discoverer was Dr. John Enck. He was just a simple scientist, maybe like us, and then he discovered this peptide-like drug. And with this drug in his hand, he knocked the door of many, many pharmaceutical companies, but they didn't pay attention. And while he was just presenting anti-diabetic activity of this molecule in one of the symposium in US, then one company, Amylin Pharmaceuticals, actually, they were really interested in the molecule, so they signed a contract with Dr. John Enck. So this drug was developed into a clinically available drug, which got approval in 2005, and now it is in the pharmacies of many countries called Byetta. This is a glucagon-like peptide analog, and which serves as a drug for type 2 diabetes patients. So there's also big and interesting story behind discovery of this drug. But Dr. John Enck, although he discovered the molecule, but he didn't made any profit, I mean, financially out of this drug. So he's still working as a researcher or director of a research center in US. And although company made a lot of maybe millions of dollars with this drug, so he didn't really make money for his discovery. And he just said that I don't expect to make like millions of dollars for this molecule, but I'm happy that I just give a gift to humankind. And I think this is the best part of our research, that we can just make such a favor for human being. So another example is conotoxin, or it was developed in ziconotide, from ziconotide actually. So the discoverer was Michael McIntosh, and his advisor, Dr. Oliveira, originally from Philippines. And this compound was isolated from a seashell called Conus tessulatus, from the venom of these organisms. Actually, this is a marine organisms, as you know. And this molecule was explored during a master thesis. So, the discovery can be even made through master thesis or a simple research. So they got this molecule, and now it is in clinical use, got FDA approval in 2004, and it is a very potent analgesic compound, sold in trade name Prialt, and it is quite potent and has potent analgesic activity as compared to that of morphine. And of course, one of the most famous clinical available drugs developed from a natural product is artemisinin. This was actually separated from a plant called Artemisia annua, and it is a very effective antimalarial drug. And WHO recommended this molecule as an antimalarial drug in 2001. And the mechanism, the exact mechanism of this drug was discovered by the Chinese woman scientist, Professor Yu Tu. And of course, she got a Nobel Prize in 2015 with her work on artemisinin. So there are lots of examples of scientists having Nobel Prize working on natural product. And another molecule that I would like to mention is ecteinacidin. This is a marine natural product, and it has a very potent anti-cancer activity. It was firstly isolated from a Caribbean tunicate extract, and then it was also later on actually identified to be produced by several bacterial strains, which lives symbiotically with this tunicate. And it was then developed into another drug molecule called trabectedin, and it is also now traded under the name of Yondelis. It's a very effective anti-tumor compound. So talking on biodiversity and this bioresources, of course, we cannot just skip microorganisms because they are very fruitful sources of drug lead molecules, of course, and drugs as well. So many, of course, active molecules were isolated from microorganisms, antibiotics in the first place, but they offer also a lot of enzyme inhibitors like, acarbose. This is an anti-diabetic drugs which are available in many countries in the pharmacies. So this is an alpha amylase inhibitor. And of course, another great discovery from microorganisms should be statin derivatives. And the first example was lovastatin, which is HMG-CoA reductase inhibitor. So, of course, we can just extend the numbers of this kind of drug lead molecules, and we can just underline once more how natural products can be good, effective, and safe molecules or candidates for pharmaceutical industry, definitely. So in the second part of my talk, I will just mention about our workflow and then my recent studies. So how we work. Actually, this is a general flow of our work. So we usually collect the plants according to traditional medicinal data, or if they have any use in like Anatolian folk medicine or Turkish folk medicine. It doesn't matter only Turkish folk medicine, because we collaborate with many groups from different countries, and of course, if the plant has any use, folkloric use in their countries, of course we also use, but we do random screening as well. So we do collect the plant and we do extraction, routine extraction, and then we test the extracts against enzyme inhibition assays in our lab, whatever available. And then we, through the large screening, we just get to know the active extracts. Then we do usually activity guided fractionation and isolation. And at every step, we just apply activity assays or inhibitory assays. Then from the active fractions, we try to identify the active compounds, sometimes through isolation, sometimes through, like HPLC or LCMS analysis, which I will give the examples a little bit later. So this kind of activity-guided phytochemical analysis is quite critical to find out the effective molecule or molecules. And then after this in vitro assays, we sometimes combine our studies with in vivo assays or cell culture methods. And then if we find out the active compounds, we do in silico analysis in the next step. So the first example from my studies is about an essential oil mix. Actually, this was formulated by a dietary supplement company in Turkey, which is called Talya, located, the company located in Antalya, Antalya province in Turkey. So they produce this kind of products, and we formulated this essential oil mix, a patented formulation actually. So this the product is called Motivate. It was designed for well-being, wellness, and memory and enhancing effect. So so since this is a patented formulation, I cannot give the whole percentages of this essential oils, but you can see that the mix consists of salvia, lavandula, rosmarinus, and two species, the essential oils actually. And then we analyzed the phytochemistry of this mix and found out that limonene was the major compound constituting of almost over 90% of this mix. So this is the major compound. So we did cholinesterase inhibitory effect and also in vivo experiments in animals. So we, in collaboration with my colleagues in Romania, actually, we did this in vivo assays as well. So we use Western adult male rats and performing some in vivo assays to measure their behavior through or directed to memory. So why maze test, and then novel object recognition tests and radial arm maze test, they were all performed. And then we also had some idea about biochemical parameters. And here you can see the results of the in vivo test that we got through Y-maze test and NOR memory activity. And also in the below part, you can see the how the essential oil mix actually improves the spatial memory, for example, in the radial arm maze. This actually reflects the data from biochemical parameters, and you can see how the essential oils positively actually affects cholinesterase and salt, CAT, GCH, protein carbonyl, and MDA levels. So all these parameters in fact, supports our in vitro and in vivo data. And since limonene was the major component in the oil mix, so we did also molecular docking simulations. And here you can see the results. The compound actually itself was more active against butyrylcholinesterase rather than acetylcholinesterase. And this came out as a paper this year in Molecules. So I would prefer from now on mostly on my works related to cosmetic research or phytocosmetics. So of course, every day we use a lot of cosmetic products, and I don't know if you ever counted how many cosmetics do you use, because or the average is almost like 10 cosmetic products every day we use. Like we brush our teeth and we clean or wash our hairs, and for women, of course, the number is higher. Like we use moisturize and some makeup products and so on. So anyway, we use a lot of cosmetic cold products in one day. And cosmetic industry is just like a pharmaceutical industry. It is in increasing trend. And of course, EU countries are the major economic drivers of this sector. Not only the number of products, but also the center of scientific innovation. These countries in EU, they really make a lot of R&D studies on developing novel cosmetics, because there is demand for that, because consumers demand for better or even premium quality of cosmetics. And the new trend is that the cosmetic products, like any other products that we use in our daily life, they should be environmentally friendly and safe, and there is great demand. So this market is fast developing due to high demand. And maybe in few last decades, we can see that there is another product group within cosmetic industry called cosmeceuticals. But considering either pharmaceutical industry or cosmetic industry, doesn't matter. We need to produce a high quality of products. And when you consider phyto-called products like phytocosmetics or phytopharmaceuticals or phytotherapeutics, the quality actually starts from field to the consumer. So there are lots of stages to provide this quality. At first you need to have a very good quality of plant, preferably grown or cultured in a controlled fields with GAP or GACP application. And of course, for phytotherapeutics or herbal medicines, GMP is required. And each step, there are lots of work like microbiological analysis, pesticide analysis, and optimization of this extraction, deodorization, and so on. There are lots of analysis and work behind developing any phytocosmetic or phytotherapeutic drug. So coming back to cosmeceuticals, as I said, these are like borderline products, and of course we know that this is between cosmetic and pharmaceuticals, and there are very certain, many certain actually definitions of these products called cosmeceuticals. That means these are also cosmetic products containing a biologically active ingredient and sometimes drug active substance, and they should have drug-like benefits different from the routine cosmetics or general cosmetics. So, of course, this is a new trend in whole world, and the share of cosmeceuticals in cosmetic market is in a big increase, and sometimes we saw that a lot of natural products or natural based molecule is being ingredient in these cosmeceuticals. And as you can see from these these graphics, these products are having better and higher shares day by day, and but the consumer demand in this area, not only cosmeceuticals but whole cosmetics, depends on the region. For example, Asia Pacific region countries, women, mostly women, of course, they demand cosmeceuticals more with skin whitening effect. Then when you come to Europe, European women demand usually products with good anti-aging effect for, you know, skin aging, wrinkles, whatever. So we do a related research in our lab, so we can test tyrosinase, collagenase, elastase, lipoxygenase, inhibitory activities of natural products or herbal extracts. And as I showed in our workflow, I said that we usually start with screening of herbal extracts at the first place, then we pick up one of them usually to go to push, you know, further experiments. So for example, when we started this work, we tested or screened more than 100 herbal extracts, and we did activity guided fractionation and did the cosmetic formulations, which I will just mention. Since I am from Turkey, when we just look into the flora of Turkey, we are quite blessed with a very rich flora, because our country has over 12,000 plant taxa. And which is good about that is that 30% of these plant species are endemic. So this is a very high endemism rate, and that means there are lots of plant species to work on. When you compare to these number in terms of global plant diversity, this is quite high, and Turkey has one of the countries with the most species, having most species in terms of biodiversity. For example, whole European continent has about 8,000-9,000 species, plant species. Turkey has already more than 12,000 species. This, of course, depends on different factors like climate change and being center of gene centers of many plant genus, and so on. So one of these plants called Cotinis cogrea, this was a part of a PhD work of my student who is now in postdoc study in Austria at Innsbruck University. So we picked up actually this plant in the first place. This is called European smoke tree. You can see the picture of this plant. This is also used as or grown as ornamental plant, and it has wide distribution through Europe, Mediterranean basin, and even central China and Himalayas. So this plant has been used in several cosmetic cold products, especially in Russia. There is one company producing baby products using this plant extract as the active ingredients. And when we looked into the phytochemistry of this plant, we can see that it is quite rich in flavonoids, especially, and phenolic acids. You can see here. So we did collect the plant from Ankara. So we prepared extracts from different parts of the plants. For example, this is flower extracts. So we did a lot of chromatographic work on this plant through its ethanolic extract. And each step we tested these fractions against tyrosinase, elastase, and collagenase. And here you can see quite good inhibition of especially collagenase than tyrosinase and elastase. So we also worked on the leaf extracts of the plant, and we did the same work again, and we got great inhibition. We observed actually great inhibition with the subfractions of the extracts. So further fractionations of the plant, as you can see, a lot of chromatographic step. We finally happened to identify several flavonoid derivatives and gallic acid methyl ester in the active fractions. But anyway, total extract is quite active against these three enzymes that I've just mentioned. And this part, only this part, we published this year in South African Journal of South African Journal of Botany. But through the thesis, we prepared several formulations out of the herbal extracts that we got from the initial screening. So we prepared two microemulsion formulation with Vitis vinifera and green tea extracts, which are quite common ingredients for cosmeceuticals or cosmetics, and then two cream formulations with Macropomifera and Cotinis cogrea. Then we created a brand name called Vitabel, and in this part we collaborated with a Turkish cosmetic company called Vivatina. Now they function in United Kingdom as well. But this has not been commercialized, I should say. This has only, in this step, not commercialized. But among these plants, we applied to patent last year in December for Turkish patent using this cosmetic formulation with a smoke tree extracts. And it is, I think, in the final stage of approval of patent. But we did a stability test for the extracts, especially for these anti-aging formulations, because it was quite effective against these three enzymes, as I've just mentioned. So the stability testing tests were performed by Vivatinal cosmetic company. Then in our faculty, we determined the particle size of these microemulsions for grapeseed extracts, green tea extracts, because these were the extracts that we prepared the microemulsions. In further step, you can see these are the results of the Cotinis cogrea, as well as three other plants that we identified with a promising effect, like Pistacia vera, besides Cotinis cogrea, Pistacia vera, Garcinia mangostana, that I purchased from Thailand some time ago. Of course, this is a Turkish plant. And Vitis vinifera, the grapeseed extract, you can see they're quite promising, showing inhibition over 50%. Then we did fibroblast viability assay using MTT method with the Cotinus cogrea, pedicel, and leaf extract, and you can see the data here. The fibroblasts were quite viable. However, when it comes to cytotoxicity, Pistachio vera extract was quite cytotoxic. It was the most cytotoxic, in fact, and although it was effective, it had quite significant cytotoxicity. Then we also tested these extracts in collagen content assay using again fibroblast, and here you can see that the most effective extract was that of Garcinia mangostana. And Cotinus cogrea extracts, they were also doing fine here. Pistachio vera, it was also okay to enhance the collagen synthesis in fibroblasts, but cytotoxic, if it comes to cytotoxicity, of course, it is not very useful, but the other extracts were quite promising. And for Cotinus cogrea extracts again, we tested them in 2 cell lines, HACAT and HUEC cell lines. They were also doing fine in this cell cultures, and this is the leaf extracts. The previous ones were flower or pedicel extracts, and this is the leaf extracts you can see here. Then we decided to formulate, to do some formulations using Niosom form with some of these extracts from initial screening. So this is Garcinia mangostana again, Pistachio vera, grapeseed, and propolis, Cotinus cogrea extracts. So we prepared Niosom formulations because Niosoms are like a new trend used in cosmeceuticals or cosmetics. So we prepared them and we observed the occurrence of Niosom particles here using particle size analyzer, which is available in our faculty. And here also we observed the Niosom occurrence, but we are able to test the extracts or pure molecules in our lab for this enzyme inhibition assays. But unfortunately, we cannot test the final product in our lab. So our conditions are not designed for final product assays. So in this step, we decided to collaborate with the cosmetic lab belonging to Austrian Drug Institute. So we collaborated with the Professor Günther Bohn. So they have a very developed lab. So they tested the Niosom formulations actually, and they compared the enzyme inhibitory activity of the extracts and its Niosom formulation. So I would like to present some of these results here. As you can see, this is the graph belonging to collagenase inhibitory activity of the extracts, and here you can see the percentage of enzyme power, and these Pistachio vera and grapeseed extracts, they had inhibitory, the highest inhibitory activity on this enzyme. But when it comes to Niosom formulations, we saw a different profile. Pedicel extract was the more effective, and leaf extracts as well. They were the most effective on this collagenase enzyme. And coming to elastase inhibitory activity of extracts, here you can see at the fact, the most effective ones belong to Garcinia mangostana and grapeseed or Vitis vinifera. And when they tested our Niosoms on the same enzyme, it was the propolis extracts and Cotinus cogrea extract as the most effective ones. We observed the different profile tyrosinase enzyme. As you can see here, it was pistachio extracts, but all these enzyme, the Niosom formulations, gave different results. So in the end of the day, we just found out that yes, the extracts, when it is only extracts, they're active against these enzymes at varying levels. But when we developed them into Niosom formulations, the enzyme inhibitory activity partially retained in the Niosom formulations. So that's why we thought the Niosom formulation is or doesn't seem to be very suitable for our extracts to formulate. So now we're working on different pharmaceutical form called emulgel. So this is this kind of formulation, or this pharmaceutical form, is also new trend in some cosmeceuticals or cosmetic products. As you can see, there are some examples. There are some products from different companies produced in emulgel formulation. So now we are working on preparing the emulgel formulations of these active extracts in order to prepare a much more effective pharmaceutical form. So another example of Turkish flora that I would like to mention is geranium species and erodium species. So they are represented by many taxa, and some of them are endemic to our country. So we did two different PhD theses on this genera, I should say. So after initial screening of all these geranium and erodium taxa, testing against tyrosinase, we picked up one species, Geranium glaberinum. They were not quite effective against tyrosinase, but this was the most effective one. So anyway, we decided to go on this species. So we did a initial HPLC analysis and showed that some two compounds actually, they were the major ones like gallic acid and ellagic acid. Then we fractionated the active extracts through HPLC and got 6 fractions, and with the most active fractions, which was the second one, so we proceeded to as Q-TOF analysis. And in this fraction, after Q-TOF MS analysis, we identified some known compounds like ellagic acid, geranium, corylagin, gallic acid, cunic acid, and some more. And also quercetin. We identified quercetin in another fraction as well. So with these compounds, we did some docking studies, like we found interactions, many interactions, but I present some of them, and we found also interactions with copper ions of this enzyme here for both compounds. And then these are the interactions with corylagin tyrosinase and quercetin tyrosinase and the active sites of these enzymes, and interactions with several amino acid residues here. We also use another in silico program, two program actually, series prediction program and PASS program. So of course, you know about these programs, I suppose. So these are to find out the correlations between the activity that you would like to test and in its database, how well they correlate, in fact. So with the PASS program, considering quercetin, we found correlation with skin whitening effect. So we already suppose that quercetin could be one of the active compounds, maybe the major one. So with the, especially with the PASS program, with the ellagic acid, we found the similar correlation with the skin whitening program. skin whitening effect with this program. So with this geranium glabellarium extract, we made a formulate a skin bleaching cream formulation. So we applied Turkish patent institute also last year in December. So I think it is also final stage of approval. Another example, coming almost to the end, that we also screen plant extracts or natural molecules against xanthan oxidase. And in this work, we worked on Eradium brandianum. This is an endemic species in Turkey, and there is no phytochemical or bioactivity studies, not even a single study on this endemic species. So this extract showed a very, very promising inhibitory activity, I think over 90% or something. So we picked up this plant to work further. And here you can see we got the ethanol extracts. We did some activity-guided fractionations, and each step we applied again xanthan oxidase inhibitory assays, and we did chromatographic work, got some sub-fractions, and then through the sub-fractions we did SCM-SMS analysis and identified several phenolic compounds like gallic acid, cunic acid, catechin, and luteolin, rutines, and flavonoids like quercetin, and also a big group of procyanidin derivatives. There was a large group of these compounds, but of course we were not identified all of these procyanidin derivatives in the extract, but of course we found out there is large amount of procyanidins here. With these compounds identified in the active subfractions, we did docking experiments and in silico bioactivity analysis, as I've just shown a while ago, and in silico toxicity. Here, as the MSMS, actually as the QTOF-MS analysis of these extracts, this is available in our faculty, so we have access easily to this machine, and it has a library press scan functionality. So we got as a kit of analysis data here in the extracts, the compounds that I've just mentioned. Here you can see the mass data. So some examples of these compounds to show their extracted ion chromatograms. For example, in this one for quercetin. For each one, of course, we got this to prove the chemical identity of these compounds. And then we did biological activity analysis using an in silico program, how this xanthan oxidase inhibitory activity overlaps with the compounds that we just identified. We used again PAS program and Swiss target production program. So PAS, which means prediction of activity spectra for substances. This is a very useful program because it has capacity to examine like more than 300 types of biological activity and also considering their structure activity relationships. And here, some of them you might be familiar with these programs, of course, and there's PA and PI values to compare. So this is the results of Swiss target prediction for each compound. Actually, we did this. So these are quite useful resources for target hunting in drug discovery. So these programs are available in our faculty. We could do this in silico analysis using different models, as you see here. So this is Vega Hub. As I said, this is available in our faculty, and we could do in silico toxicity and in silico biological activity analysis here. So PI should be bigger than PA should be bigger than PI values to make sure that the activity we expected actually matches. So toxic effects also you can just, of course, predict if it is if the PA data is bigger than 0.7. So for each compound, we did this probability testing for this enzyme. Here you can see PA and PI values. Then with the detected compounds, which some of which could be effective against this enzyme, we proceeded them to docking simulations, as you can see here. So anyway, for each compound we did that, but due to time limit, I'm not able to show all of them. So in the last part, or in the end of my talk, I should acknowledge the people or collaborators who worked with us. Of course, in the first place, Professor Günther Bohn, Thomas, and Ronald from Austrian Drug Screening Institute, and then Katie, we call her Katie, from Konkan University in Thailand. And for our Niosom and Emuja studies, we collaborate with a young scientist from my faculty, Pharmaceutical Technology Department, Emre. So our project team is usually consists of women, as you can see. And in the below, you can see my colleague Gökçen, who is doing the docking simulations, and the others are also working working together in different projects. So this is Hayat company, which is located near to Istanbul. This is a big chemical plant, as you can see here, a big chemical company. So we also collaborated with them, with their R&D laboratory to, in some part of this cosmetic research, they also helped us, especially with the cell culture assays. So in the very end of my talk, I would like to thank you all for your patience, for listening to me. I hope you enjoyed my talk. Thanks a lot for giving me this opportunity.
Thank you, Dr. Ilkay. It was certainly a very interesting presentation and talk. The floor is open for question and answers. Do we have any questions, please? Kindly unmute your mic if you want to ask a question from Dr. Ilkay.
Hello.
Yes.
I have a question.
Yes.
When you put your sample on LCMS and you got many peaks, then how can you sure that this peaks belongs to that one components or that one phytochemicals after LCMS analysis?
So we of course compare the mass data, basically.
Are you sure that mass data gives?
Yes, of course. I mean, in that sense, we have also what we call a standard of this compounds in our hands, so we can get the mass data of the standard compounds and compare mass data to each other. So we can make sure in that way.
No, are you sure this way is enough for the full detection or identification of that component just on the basis of LCMS peak?
Well, we have library, and we of course compare with the literature. We compared with the MS of our standards, and these are very well-known compounds. Like, you cannot skip. If there is a new compound, of course you can suspect and you can do more work. But these are the compounds that we detected. They are already very well-known ones, like quercetin or gallic acid, or so on.
No problem for quercetin. Usually, for example, if I personally hit any peak of LCMS, then thousands compounds appear even of a similar class.
Yes.
They have quite small difference. Then how we just sure that this peak belong to that specific phytochemicals?
but we do it like by HPLC, we identified, and we do LCMS, and this is Q12MS we use.
Okay.
So we are sure, I mean, I don't know, how can I convince you?
May I ask a question, please?
Yes, Asad?
Yes, Professor Ilkay, pleasure hearing you again and enjoying this very informative lecture.
Thank you.
Thank you, really, thank you. It was excellent. One of the things I admire very much about the research you are doing is it's not only a basic research. You take it further towards patenting. You are trying to find a place for you as a scientist in scientific and commercial community, which is very good, which is in fact all of us should really think to do, because this is the only way that through which we can really get connected to people and benefit humanity. You have said during the lecture that you have some of the extracts patented, and you have some of them maybe now even produced in a form of pharmaceutical product. I want to know how much easy and difficult it is to patent a natural product. And because in fact, this is very important part we should also consider. Thank you.
Right. Thanks for a nice comment. And it is also nice to talk to you even online conference. So anyway, actually, of course, it needs personal perceive definitely, but our university also encourages us to apply patent, because I think most of the universities in the world, or in considerable number of universities in the world, they have patent office like we have here. So our university patent office, they encourage us and they helped us how to apply to patent institute. You know, they give us a very nice tutorial about that. And, you know, it also needs some financial support in that way for application to any patent institute. And our patent commission, if they believe in your work and if they agree that this may have a good commercial output, then commission decides to support the patent application. Then they pay for these steps. So that's why in our case at our university, it is quite easy to undertake this kind of patent application. So in that sense, last year I have 4 patent applications, and I have 3 already registered patents. I didn't mention about those here. Like we have one Turkish patent already in 2017, another one European patent in 2018, and US patent in 2019. So I'm also expecting to get these patent applications to be positive this year or maybe early next year. So you are right. This needs some encouragement, probably not only by yourself, but also by your institution. So this is my story. I don't know it is how general it is, but this is my story behind applications, patent applications.
Okay, thank you very much, Ilkay. Thank you. Thank you.
Dr. Ilkay, we have a question from Dr. Professor Reza Tanayev from Iran. Dr. Reza, can you please ask your question?
Hello. I was wondering, how far do you actually get this patent? Do you actually prepare everything and patent it and then submit it to the commercial industry, or you start to present your things to the commercial so they can provide you with money and financial support? Because finding a company and convincing them in order to actually start from the beginning to pay for their research is very difficult, at least where I live. So my question is, how far do you actually get your patent before you actually could submit it to the industry and get the financial support for it? Thank you.
Thank you too. Considering my patent applications in my hand at the moment, I didn't apply or I didn't talk to any industry company or sector company at the moment. So these are still at the stage of applications. But after the application is finalized, and if it is positive, of course, we can start to talk to companies. But in the past, as I just mentioned, I had 3 patents on derivatives, which have also a skin bleaching effect. So for those 3 patents, for example, one of the colleagues who were in the patent team for that work, she founded and established a company, a cosmetic company called Koji Cosmetics. So although we were part of this patented work, so she developed these formulations into cosmetic products, and she has kind of started to sell them now. But considering my patent applications, I don't know how they will be worked out. We just need to see when they are available as the real patents. But as Asad asked a while ago, the process was even the application to any patent was difficult for us too. It was like very hard work. You need to do a lot of paperwork, and as you mentioned, yes, you have to work good amount of money and etc. But this patent office founded several years ago in our university, now they make everything on behalf of you, so that really encourages you to apply for a patent. They also work with the patent offices outside of university, that companies, they do everything on behalf of your work to the Turkish Patent Institute, they pursue it. So that's why we are now more eager to apply to patent. But of course, as you say, I would like them, at least one of them, to be commercialized. So when I have the patent in my hand, especially with this skin bleaching formulation, for example, I can talk to the cosmetic company, and if I can convince them, of course. And with the other one that I mentioned, that led to establishment of a cosmetic company, Koji Cosmetics, which was established by my colleague. For example, TÜBİTAK, Turkish Scientific and Technological Research Council, which funds the major grants in Turkey for researchers. So they were aware of our work and patents, so they contacted us, and they were in between us and a cosmetic company called Flormar in Turkey. It is a famous brand in Turkey. They also do a lot of export. So now we are dealing with them to commercialize this product. They want to buy the license. So this is how it goes. This is what I can tell. Thank you for your question.
Thank you, ma'am.
Okay. Thank you too. So we are done?
Yes, ma'am.
Okay, so should we say goodbye?
I think Khazima has some connectivity issue, so I would like to thank on behalf of ICCPS and Comstech a very Great. Thank you to Professor Dr. Ilkay and all the participants, and all the registered participants will have e-certificate received. And with this, I would like to thank you all. Thank you so much.
Thank you.
Thank you, Professor Ilkay.
Thank you, Dr. Hena, and thank you, Dr. Ilkay, and to every of our participants.